Okay. So in last lecture, I sent you two links. Have you watched the video? Actually, two videos.
Yes.
Yes.
Yeah. So that's about protein. It's about the protein design. So you start from a sequence. Actually, you start from a desired structure, and then you use some strategy to design a sequence, and that sequence finally can fold into the structure you want. So this is the one of the dream for for us, for the researchers. If we can design a protein using some sequences, um, it can do a lot of things. Like you can design some protein to fix some issues in the human body, to cure some diseases, or you can also design some peptide which can be used as drugs or something. But that's about single protein. So today we're going to talk about multiple proteins. Let's start from two proteins. So protein-protein interactions and docking algorithms. But before that, we can discuss a little bit more about single proteins. So in computational biology, there are several big areas. For example, the first one is protein folding or protein design. So the videos I sent you, it's from Dr. David Baker, who's a very famous scientist in this area. They design proteins, and they also develop computational tools to simulate the protein folding. Protein folding means, okay, let me maybe show you a picture. There are too many images. I can't just use one picture here. So for example, if you have a sequence or a peptide, so this is a sequence of amino acid or residues. If you have the sequence, can you predict the structure of the protein? Because a protein is essentially an amino acid sequence peptide. So if you have this peptide, how do you use computers to predict the structure of the protein? This is the very difficult task. This is number one area. So protein structure related research. So then the second big area is protein-protein interactions, or let's say docking. In protein-protein docking, what you want to do is, if you have two proteins like this, the orange and blue one, you know they have interactions. In experiments, you can measure the structure of the protein A and protein B. However, in many cases, even you know the structure A and the structure B by some experiments, but in many cases, you don't know the complex structure because it's very difficult. I don't know the details of the experiment, but I know it's too difficult sometimes. So one task for researchers in computational biology is to predict the complex structure of protein A and protein B. This is also very difficult because you can imagine for protein A, it has so many orientations. For protein B, it also have so many orientations. So they can bind to each other in almost infinity number of positions. So in your algorithm, you need to do a search and finally identify which position is the best position for the complex Thanks. Okay, I hear some voices. Now let me see. I'll mute Javier. Okay, now the voice is gone. The noise is gone. So that's protein docking. There are also some other big areas like drug design. For example, here. In drug design, computational biology drug design method use some softwares to screen some drugs, or sometimes even develop some drugs and do some simulations to identify which drug can bind to the target, can, for example, inhibit maybe some misfunctioned protein, or increase the activity of some proteins or something, which is related to the disease. So this is also a big area. But here today, we're going to talk about the protein-protein interactions. For those three areas I was talking about, they're very hot. The first one is the single protein structure prediction. The second one is this protein-protein interactions prediction, or docking. The third one is the drug design. Because there are so many groups or people working in those three areas, so there are some even worldwide competitions in every year. For example, CASP, protein prediction. So this is a competition. This is an organization which organizes the competitions of protein folding. So, for example, in every year there are so many new structures determined by some experiments. Some of them are using NMR, others using X-ray or cryo-EM or some other experimental method to determine the structure. In order to verify the existing algorithms which are working on protein structure predicting, they organize this competition. So every time when there's a structure available, sometimes the organizers will kind of blind this structure from the world, so they don't release the structure. Instead, they give the sequence to the groups who want to attend this competition. So those groups who develop softwares, they attend this competition, they get the sequence, but they don't know the structure because the structure is blind. So nobody knows the structure except the experimentalist and the organizer. So every team or every group in this competition, they will predict the structure using their algorithms, and then in a couple of months they submit their results, and then the organization will do the comparison between the predictions and the experimental structures, and then they rank. For example, this is CASP 14. So in each round of the competition, they do a rank to see which group or which software is the best to predict the structures. Okay, let's see. Where is the results? Go scope, target format results. Okay, maybe this is too new. Let's see previous competitions. results, assessment. Okay, I forgot, but there's a link. When you click into the link, you will see the performance of each group or each software. So that's CASP. Another competition is Capri. Let's see, Capri protein dot interaction. This is another organization. Say they organize the competitions of protein docking algorithms. So it has been over 40 rounds. So when I was a PhD, I attended several competitions. If I remember correctly, it should be something like around 1920 or something. Participants. So in this competition, the organizer give you two proteins. They know the structure of the complex, but all the team members they don't know the complex structure. They only get the structure of single proteins, and they will predict the structure of complex. In a month, you need to submit your results to the organizers, and then the organizers will grade your prediction and then give you a rank. Participants. So you can see there are so many participants, like over 30 groups in each round. But later on, in recent years, there are even more teams. And I forget how to see the results. So in each round, the result is open for public. So you will see which software works the best. But of course, we can't judge a software only based on a single round of their performance, or even based on a couple of rounds of the competitions, because some softwares or algorithms, they may have their advantage. For example, some softwares they might be good at predicting very flexible proteins, others are good at rigid proteins, or some are good at protein bind to RNA or DNAs. So different software may have different advantage and disadvantage. But here, I just want to show you some background of the computational biology, especially in the algorithm or program developing directions. That's a very difficult direction. So because a lot of times you have to compete with the best groups in the world so that you can be recognized. Also, there are also some competitions for drug designs. It's similar. So the organizers will give you some target, and then you design some drugs, and in perhaps a couple of months, you submit the result, and the organizers will rank which algorithm or which team is the best. Okay, so in this lecture, we're going to talk about protein-protein interactions and docking algorithms. If you have any questions, please feel free to stop me. Okay. So first, background of protein-protein docking. This is the background of protein-protein docking method. So in a living cell or in human body, on average, each protein interact with 3 to 10 other molecules. For example, you can see this is a network. Each dot means a protein or some other molecules like ions or DNA, peptide, some other molecules. So each protein will interact with at least 3 to 10 other molecules, including other proteins, or sometimes even the same type of protein with itself to fund its function. So biology functions are always in protein-protein interactions. So you can imagine if a protein is isolated, if it doesn't interact with any other protein or molecules, then that means this protein is kind of useless. It doesn't have any function. So the protein has its function by interacting with other proteins. The most commonly recognized functions are shown here. For example, signal transduction, catalytic reaction, and immune systems, and many other functions. So most of the functions, actually, most of the biological functions of proteins are perhaps performed by protein-protein interactions, because there are too many different types of proteins in living cells. So here it shows one protein-protein interaction. This is perhaps one of the most famous protein complex, 1BRS. If you remember, we have been using this 1BRS.pdb as our example in the previous Linux, I forgot, Linux or Arc. Maybe in Arc, when we study Arc, we use this PDB as our example. So this PDB or this complex, it contains two proteins. One is Barnes, the orange one. The other one is Barstar. It's an enzyme. The Barnes, it's an enzyme, and the Barstar, it's an inhibitor. So this 1BRS is one of the earliest determined complex structure by experiment. And its interaction, the interaction between Barnes and Barstar, it's very classical. You will see electrostatic interactions and hydrogen bond and salt bridge, almost everything at the interface. So it's a very classical example. So many algorithm takes the 1BRS as a classic example to do tests. For example, when you first develop a protein-protein docking algorithm, most probably you want to use some proteins such as 1BRS to test your program at the beginning stage. So in computational biology, there are a lot of groups that are working on developing new algorithms to predict the protein-protein structure. For example, for 1BRS, if you don't know the complex structure, instead, you just know the structure of the Barnes and the structure of Barstar. So can you use an algorithm to predict the structure? The motivation of doing this is because in experiment, the complex structure is much more difficult to obtain compared to a single structure. So even in the PDB bank, if you do a search, you will see most of the available structures in the PDB bank, they are single structure of proteins or molecules. For complex structure, it's very limited number. So one goal is, let's use some algorithms to predict the structure of complex. Okay, so that's our task. If you have a single protein structure and another single protein structure, and in experiment or in some evidence, you know those two proteins, they bind together to perform their function. However, nobody knows their complex structure. So our goal is to predict the complex structure. It seems like a very complicated task. However, um, it might be easier in many cases than you expected, because in a lot of cases, when a protein bind to another protein, in many cases, the structure of the two proteins, they don't change too much. So in this case, it's called rigid body protein docking. That means, let's say the two proteins, one is receptor, the other one is ligand. So the structure of receptor doesn't change too much before and after binding to the ligand. And the same thing to the ligand, the structure doesn't change during the protein interaction. In this case, we can use some we can use some classic method to do some predictions based on geometric complementarity, because due to the analysis of existing protein-protein complex structures, we will find that actually the protein-protein interactions, it's very smart. They match each other very well in most of the cases. When I say match them, they match each other very well. We mean geometrically they match each other. So for example, let me see if I can draw pictures here. Sort of tools. Okay, I'll try to draw some pictures.
Read the shape.
Shape.
You can try the shape.
Insert.
No, picture. Down, leave it down. The right, to right, to the right.
Oh, okay. Thank you. Shape. Draw. Okay, now I find it. Draw. Okay, thank you. Now let's see. If you have one protein in a structure like that, if the other protein is in a structure like this, so most probably they bind in this situation, because in this case they match each other in the best way. Otherwise, if you do a rotation, let's say this is receptor, this is ligand. If you do a rotation to the ligand, then there's no way for the ligand and receptor to match each other in the best way. So this is called lock and key. Lock and key. This is called lock and key model. And it is true for many protein-protein complexes. And many famous widely used protein-protein docking algorithms, they're designed based on this lock and key algorithm. They will first treat two proteins to be rigid body, 3D, let's say, structures like Legos. And then they will do a lot of rotations and translations and do a search to see in which case they match each other the best. And the best case is the final prediction for their algorithm. So this is the number one rule, geometric complementarity. Okay. Now here, let's see two classic examples. First one, 1AVX. What's this? Okay, so this is a another. Let me see. 1AVS. I forgot what it is at, but I believe this is inhibitor and enzyme. So the blue one is an enzyme, but the white one is inhibitor. I forgot. 1AVS. Maybe we can do a quick search. 1AVS-X. Yeah. Anyway, so it has some biology functions, but it's enzyme and inhibitor. So this is the first type of protein-protein docking. This is the structure after docking, or this is a complex structure determined from the experiment. You can download this structure from PDB Bank as shown here. That's complex structure. It's very difficult to obtain, but luckily it has been determined by some experimentalists. If you have the complex structure, that means more likely you have already have the single structures of this enzyme and inhibitor individually, maybe long time before you get the complex structure. And this is the case for 1AVX. We also have the structure of individual enzyme, which is shown in yellow. And also we have the single structure of the inhibitor, which is shown here in pink. If you align the single structure of the enzyme and the inhibitor to the complex structure, you will see there's only small scale conformation change, means it's very rigid. So this is a rigid body docking. And in this case, it's not easy. It's relatively easier for algorithms to do some predictions. In another case here, 1FAK. So for example, this is a complex structure of another two proteins. If you align their single structures before docking or before interaction to the complex structure, you will see here the blue and white one, they are the complex structure, and the yellow one is single structure, and the pink one is single structure. You will see for let's say this is receptor. If we call it a receptor, you'll see the receptor undergoes a big or large scale conformational change before or after the interaction. You see the yellow structure before the interaction, here, this loop, and even some other detailed structures are quite different from the complex structure. So for this type of protein-protein interactions, so far as I know, there's no very effective method. If you have a structure which undergoes quite flexible conformational change, then for all the existing protein-protein docking algorithms, it's difficult. So in today's lecture, let's focus on the easier case, which is small-scale conformational change cases in the protein-protein interaction. Okay, in this case, as we said, if there's no large-scale conformational change, most probably we use lock and key model. Means we are trying to find the geometric complementarity. Okay, so now first, if you want to find the geometric match, then at least you need to represent your protein in geometry in your computer. So you have seen the PDB file. In a PDB file, if you remember, you have a lot of record for atom. So for each atom, it might be hydrogen or carbon or oxygen atoms. For each atom, you have the X, Y, Z coordinates. So if you output or visualize all the atoms, you may see something shown here. This is a visualization of a PDB file. So each atom is shown individually, and if you show them together, you will see the whole structure of the protein in this shape. Maybe some of you haven't tried some visualization software, but next week, I believe Wenhan, she will give a lecture about how to use VMD and the Chimera to visualize proteins from a PDB file. Then you should be able to do it. But at least you know from the XYZ coordinate of all the atoms, you will get a structure like that. This is a visualization in Chimera. But in docking algorithms, you need to represent this structure in your software or program. So the easiest way is you can map the protein into points, grid. So for example, you have a protein which is in this shape. Of course, it's 3D, but here to simplify the example, I plotted in 2D. So now you have protein B in this shape. What you can do is you can make a grid, n by n grid, and then you map the protein B or centered the protein B in the center of your grid. And then you can use some functions or some algorithms to identify if a grid is occupied by B. If it's occupied, then you can assign a value as 1. If not, you assign a value of 0. And then finally, in computer, you will get some protein in this shape, or an array in this shape. In your computer, it's a 3D array, but here I just show a 2D case. So you have some white space, that means 0 values, and here it's a blue, means it's 1. So that's the easiest way to represent your protein in a 3D grid. Okay, do you have any questions so far? Okay, then let's continue. This is protein B. Okay, if we write the equation or write it in a formula, what you do for protein B is you use a discrete function f, b, lmn. Lmn are the indexes for x, y, z directions of the grid. And the value of f is either 1 or 0. 1 means this grid is in the protein. 0 means this grid is out of the protein. In this case, you define x, y, z, or LMN 3D grid, and you get the shape of your protein B. And of course, the accuracy of your representation of your protein depends on how fine is your grid. If you use very coarse grid, then the shape of your protein is also coarse. If you use a very fine grid, then the shape of your protein could be very detailed. This is protein B. Now let's see protein A. For protein A, we use a different strategy. Now let's first look at protein B. For protein B, you have 1, 0. So blue means 1, white means 0. But for protein A, we make it a little bit more complicated. So outside the protein, we still assign 0. On the surface of the protein, we assign values of 1 here. But inside the protein, we assign value of -15. Why it's -15? It's been tested by some algorithms. So -9 to -15, it's the most effective value for the docking algorithms. So now this is the function of FA and FB. For B, it's easy. Inside the protein, it's 1. Outside, it's 0. But for A, it's a little bit more complicated. On the surface of the protein, the value is 1. Outside the protein, the value is 0. In the core of the protein, the value is -15. So now you have represented your protein A and protein B in your computer. They're just two arrays, of course, three-dimensional arrays. Using these two arrays, you will do some amazing work, which is shown here. It's a very smart idea. Now let's see how does the protein docking algorithms do. Okay. First, let me fix the typo. Okay, now let's see how to translate the protein and get score or matching score for different binding mode. So remember, our task is trying to use the lock and key model to find at which orientation and which position those two proteins match each other in the best way. That's our goal. Or our goal is trying to find the best geometric complementarity. Then in order to do this, what we need to do is, of course, we need to translate and rotate one of the PDB to do search, see at which position they have the best match. then you can imagine if you are the developer, at least you need 3 important functions in your algorithm. The number 1 is do translation. You need to move it up and down, move it left and right, move it backward, forward. So you search in a 3-dimensional coordinates. This is number 1 function. Number 2 function is do a rotation. You need to rotate one of the PDB in 2 pi. But in a 3-dimensional case, besides rotating it in this direction, you also need to rotate it in another direction in this axis. So you need to do a rotation of pi. So that's a 3-dimensional rotation. That's number 2 function. The 3rd function is after a combination of translation and rotation, you need to calculate the match of the 2 proteins. For example, you have the first protein here, you translate the second protein to here, then you need to know how well they bind. And then in another case, you move the other protein here, you need another score to quantitatively indicate which case is better, in which case they bind to each other or they match each other better. So that's the 3rd function. Try to find out a quantitative value or score for a match. So what we can do here is, for protein A, let's keep it fixed. It doesn't move at all. We only move protein B. So first function, let's do a translation function. You can move protein B. In a 2-dimensional case, you can move it left or right, move it up and down. In a 3-dimensional case, of course, you need to add 1 more dimension, which is moving it forward, backward. And in some cases, okay, every time when you move it in a small step, actually the step size is the same as your grid size. For example, this is your grid size. So each grid is very small. So in your movement or translation, you also translate it grid by grid. So each time you translate it by a very small step. Every time when you translate it, we calculate the match score to see how good they match each other. For example, in this case, A and B, they are separated. Then the score, by some algorithm, the score will be 0. Why? Because we use such a function. We use a correlation of FA and FB. Let's say the correlation of A and B is FC. C means correlation. Alpha, beta, gamma. So alpha, beta, gamma is a vector. Means if you move protein B by alpha, beta, gamma vector in X, Y, Z directions. So this is a vector between A and B. So once you move B to this position, now let's do a calculation of FA times FB. Actually, it should be dot product. Okay, let me see if I can. Okay, it's difficult to do modifications. So here it should be a dot product, not a cross product. So what does this mean? It means, for example, you move B here. Now let's do a calculation or a product of A and B on each grid. Now let's look at this grid. In this grid, what's FA? What's the value of A? It's 0 because it's out of protein A. At this position, what is the value of FB? It's also 0 because it's also outside the protein B. Now let's take a look at this grid. In this grid, it's inside protein B, but outside protein A. So FA equals to 0, FB equals to 1. But if you do a product, FA times FB, still the value is 0. Now let's see this grid inside protein A. Inside protein A, the FA is negative 15. And this grid, the grid value of FB is 0 because it's outside protein B. So if you do a product of FA times FB, still it's 0. So in a big space, let's say this is a very big space, it has a lot of grids, 1 by 1 or n by n. So for each grid, you do the FA times FB. You will figure out at the end, you get a 0 value for each grid. If you sum them up, if you do a sum of all the product of FA and FB, so finally you will get a 0. So if the correlation equals to 0, it simply means A and B, it doesn't have any contact. Now, let's look at another case. If A and B, they are kind of bind to each other, but no penetration, then let's look at this grid. For this grid, FA is 1. FB is also 1 because it's on the surface of A. It's also inside the protein B. So FA is 1, FB is 1. So FA times FB is 1. And all the grid which are on the interface of A and B, they result in a product of 1. And for all the other grid, for example, here, the product of FA and FB is 0. Here, FA times FB is 0. So if you do a sum of all the FA times FB in the space, you will have a positive value. So positive value means A and B, they have some contact, or they interact with each other. They have some interface. FC equals 0 means no contact. We have explained it in previous slides. Now let's see the third case when they have penetration. That's interesting because first let's look at the interface here. For this grid, FA is 1, FB is 1, so FA times FB, it's 1. It's a positive value, but it's 1. But for this case, it's in the core of protein A, it's also in the protein B. If it's in the core of protein A, it means it's negative. FA is negative 15, and FB is 1. If you remember the rule we used for assigning values to FA and FB. So inside A, it's negative 15. Why do we use negative value and negative 15, which is much bigger than 1? Simply because we're going to use this to do some penetration score. So if one grid is in this situation, the FA times FB is negative 15. So if you do a sum of all the FA times FB for all the grid, you will have most probably a negative number because the penetration, for penetration situation, the penalty score is is a huge score. It's -15. So in this case, it's your correlation function is negative score. Now, let's look at all the three situations. When A and B are far away or they have no contact with each other, the correlation score is 0. If they have some match, then the correlation score is positive number. If they have penetration, then or overlaps, then the score is negative. Okay, now we have introduced the score. So next, the thing is, let's just do a search by fixing A and moving B at in all the space surrounding the protein A. Do the three-dimensional search. And at each position, let's calculate the correlation score. If we see it's 0, we know there's no contact. If we see it's negative, we know it's overlapping with each other. If we see it's positive, we know that might be a good match. And then for each position, you will have a score. This is for translation. But you can imagine it's a very time-consuming process because you need to. It depends on your grid. If you have a lot of grid, then this calculation takes a very long time. However, we can use fast Fourier transform or FFT algorithm to let's say accelerate the search. You don't have to really move the protein B grid by grid and do the search in space. What you can do is you use FFT algorithm to do this thing. So it will speed up this search significantly. I will not introduce this FFT here because that's involving too much mathematics. Anyway, you just take the FFT as an acceleration tool to accelerate the search. And then you can do a rotating. You can rotate B in Euler angles, which is 2 pi by 2 pi by pi. It's a three-dimensional rotation. For each rotation, you try to find the best match by translating the protein B in space with protein A. For example, if you rotate protein B in this angle, maybe you can find the best match for A and B is in this case. If B is in this orientation, the best match is here. If B is in this orientation, the best match could be like that. And of course, you will have scores for each match. And in this case, they match each other the best. It's like lock and key. So the interface is the largest. Largest interface results in largest positive number based on the algorithm we explained here. So when you look at the score, you immediately find out in what position A and B they match each other in the best way. Okay, so that's the algorithm of protein-protein docking, especially for rigid body protein-protein docking. It's very similar to image recognition. Maybe some of you, for example, I know Yixin has been working on some image processing or image recognition. So in image recognition directions, everything you need to use, almost everything is related to fast Fourier transform because it can accelerate the kind of the search algorithms. So in protein docking, you're trying to find out the best match of protein A and protein B. It's another type of image recognition. Actually, for example, in this case, you're trying to figure out, okay, A, it has a surface in this shape. So if you find B, it has a similar surface in this shape, that means they can match each other in this interface. So we're using the image processing algorithm based on FFT to do protein-protein docking. After rotation and translation scans, we finish the scanning of 6-dimensional space, and you will have a lot of different positions. And for each position, you have a score. You can rank the score and pick up the best scores. So that would be your best predictions of protein A and B. So that's all for the basic protein docking algorithms. So do you have any questions so far? Okay. Do you all lost? Are you lost?
No question, thank you.
Okay.
It's very clear, Dr. Lee.
Oh, okay. Thank you. Okay. Let's see. Is there anything interesting to you? Let me quickly go through the slides quickly. Okay, so far what I'm talking about is basic protein-protein docking algorithms. But in the world, currently there are so many different protein-protein docking algorithms. Let me show you something. I listed several docking programs. They're not all the algorithms, they're just some of them. Why do I list these? Because these are the algorithms which have been used in Capri competition, and these are the most successful docking algorithms. So different docking algorithms add different features to the basic docking method. For basic docking method, we do the search for the best geometry match. But for example, when I was PhD, I developed a docking algorithm, and I added some other features, which is called ASP, atomic sublution parameters. It sounds like complicated, but the basic idea is we know that besides geometry match, there are also many other matches. For example, electrostatic matches. We know, let me see, it's difficult to find a picture, but okay, maybe I'll draw something here. We know that, for example, besides the geometry match, we know if there's a lysine, it prefers to match to maybe glutamic acid, aspartic, amino acid, or maybe sometimes, yeah, most of the time it's matching those two types of residues because it's positive. These are negative. If we see on one side you have a glutamic acid. Then you know most probably it likes the lysine because lysine is positive, glutamic is negative. Glutamic acid is negative. It prefers lysine or arginine because it's also positive. Then you can try to find some scores on different amino acid or atoms, so that when glutamic acid see lysines, it has a good score. When glutamic acid see perhaps aspartic amino acid, it will give you a negative score because they are the same charge. So in this case, instead of assign value 1 to A and B, I try to assign different values on the surface. Some of them are positive, some of them are negative. It's complicated matrix. I didn't develop this matrix. This matrix describes the matching score between different types of amino acids and atoms. When they meet each other, are they in a good score or bad score? Are they positive or negative? It's developed by many other groups. So I just use the matrix and try to implement this kind of so-called atomic salvation parameters into the basic docking algorithm and try to improve the basic docking algorithms. And then we use this algorithm to participate in 2 rounds of the Capri competition. So this is the Capri competition. This is the target, and this is the result my team predicted. So it was very good. So there are so many different types of docking algorithms. So this is a rigid body Brownian or Monte Carlo based algorithms. For example, smooth stock. Okay, it uses Coulombic and Salvation energies plus lock and key model. What's this? This is Moffitt. It uses geometric FFT, that's the basic. And then scoring by weighted geometric complementarity. So that's their perhaps new method, weighted geometry. So in our geometric method, it's uniform, but maybe they have some new method to weight the geometry complementarity. Guided docking. This is some docking algorithm based on charm force field. It can do some flexible refinement in some cases. Gap dock. Scoring based on shape, correlation, and buried area. Okay, so anyway, so there are so many different types of docking algorithms. So first, they based on geometry complementarity, then they add some features to improve the docking algorithm. So this is the basic knowledge of docking algorithms. I know some of you may need to do some project for protein-protein interaction or protein-protein docking. So currently, one of the best protein-protein docking algorithm is ZDock. If you go to the ZDock server, you input your first protein, then second protein, give your email and submit your job. So after some time, it gives you the complex but it doesn't give you only one complex. It might give you 2,000 complex, and with the rank, you will see the top 1, the top 2,000 predictions. So for example, ZDoc, it's based on the geometry complementarity plus several other factors like electrostatic and other features I forgot. So when you combine more features, you may get more reliable algorithms which can better predict the structure. Okay, so that's the basic idea of protein-protein docking, especially for rigid body protein-protein docking.